Thank you, Jill, for the kind introduction. And first of all, I would like to thank the organizers, especially Alvaro Villon, for inviting me and giving me this opportunity to come to South Africa again. So this is actually my second time, and I really enjoy it, actually. What I would like to do today is give you a little bit an overview about the research topics that I was working on the last couple of years, and that involves polyphenols as biogenic additives. It's a new term, and I will explain it to you very shortly, and the polyphenols role of polyphenols as value-added products. Now, you all know the saying, an apple a day keeps the doctor away, actually. And the question is, why is this so? And you know that we are all told we have to eat enough vegetables because eating vegetables is really good for us and it's very healthy. The reason for this is that especially vegetables contain polyphenols and in particular flavonoids. And these flavonoids, if you look into the literature, there are many papers dealing with anti-cancer activities, heart diseases, obesity, Alzheimer's disease, metabolic diseases, depression, and everything. That means, obviously, flavonoids are good for good and against for everything, basically. The question is, if this all would be true, then we would have the perfect cure for almost every disease. And since we all know that this is not the case, something is not right. And I would like to point today to some facts and especially to some challenges in flavonoid research. And some of the challenges in flavonoid research are, first of all, if you look at the structures, they have a really large diversity. And you have, if you look at dietary sources, you also have a very large number from vegetables, fruits, and so on. And if you further look into the literature, you see that there are many in vitro studies out there, and the bioactivity, especially of parent compounds, and I come to this in a second, is studied. And then, if you further look that in vivo studies very often use high doses, unrealistically high doses. They have no relevance at all. They are mostly supraphysiological. And very often you have a lack of in vitro and in vivo correlation. So that means you find great in vitro data, but you cannot correlate this to any in vivo activity. So this is really a pity. And then on the other hand, this sometimes makes me really angry, that you have a persistent focus of research into activities of flavonoids like antioxidant activities and so on, which is pretty much predictable nowadays. On the other hand, and Jill mentioned this already, you have a limited amount of studies investigating the bioavailability, metabolism, and pharmacokinetics, especially of flavonoids. And if you think it's true, if you really think, okay, you know, flavonoids are the magic drugs, you would need large amounts, but where should they come from? And there's a low production efficiency from plants and chemical synthesis. So this is also a very important point that needs to be addressed. Now, what I would like to do now is show you some work that we did, some in vitro work, and then where we failed in vivo studies and how we addressed the problems, and what I think might be a solution of this. Now, a couple of years ago, I was working on a project. It was together with a company, and what we wanted to do is we screened several flavonoids, several plant extracts actually, and then identified some flavonoids for xanthine oxidase inhibition. And so it's a relatively easy in vitro assay, very easy enzyme assay. So we screened several compounds, caffeic acid derivatives, but also flavonoids. And there are different groups of flavonoids, flavones, flavonols, flavonones, and so on. And we found that especially luteolin, one of these compounds over here, is very active. It had a very low IC50, 1.5 approximately, and it was more active than our positive control allopurinol. So this was really promising, so we thought this is great, so maybe we can continue this from here on and can use luteolin as basically a template or something to develop natural product-based drugs. Now, then we did an in vivo study and completely failed. So we started with low doses. These are already 16 milligrams, 30 to 50, 100. So we went up and up and up, and we didn't see any in vivo activity. So maybe I should say that if you test for in vivo effects, you measure the uric acid, plasma uric acid actually. And I have to say we did a rodent study, and rodents usually do not develop gout. So you have to induce it because rodents have another enzyme, uricase, which metabolizes uric acid into allantoin, which is water soluble and can be excreted. Now, we needed to give uricase inhibitor, and this was potassium oxonate, so we give this, and you see the uric acid levels went up, and then we gave this together with our positive control allopurinol, and then with these really huge doses of luteolin, and nothing happened at all. And then we decided, okay, let's check other flavonoids like camphorol, arbugenin, quercetin, and so on. In the same dose, let's just take 50 milligrams and let's see what happens, and we saw nothing. So this is what I'm saying. So you have this lack of in vitro, in vivo correlation. And then we ask the question, okay, maybe it's because of the pharmacokinetics, maybe of the bioavailability of luteolin. Let's do a pharmacokinetic study. This is what we did. I'm not going into details here, just one number that I would like to point out, and this is the bioavailability of luteolin. After oral administration, we determined it as just 4%. That means 96% are somewhere gone, actually. We also measured how much of the luteolin, the parent compound, but also the conjugates, get excreted. These are the urine levels, actually, and we found that after oral treatment, maybe 1%. Here you have for the conjugates 4% and so on. And after IV administration, also you find really low levels in the urine. So that means over 90% disappear somehow, but where does this compound, where does it go? It cannot simply disappear. Probably at the same time when we did this study, I found a very interesting study in the literature published by Mullen and co-workers, and what they did is they took radioactively labeled quercetin 4 glucoside, and then also gave it to rats and then followed its way through the entire intestine, and so you could see already that there is a high metabolization rate. It starts already in the stomach, but that a relatively large amount of quercetin actually enters the ileum, but also here the colon, and is metabolized by gut bacteria to small metabolites like hydroxybenzoic acids or phenylacetic acids, so really small and common known metabolites. Now, so you see, I mean, you have from the parent compound, you just have a really small amount actually. So that means if you would do a pharmacological study, right, you actually would have to work with the metabolites and not so much with the parent compound, because most likely those compounds probably would have an effect actually. So I proposed this study to funding agency. I said, you know what, the compounds, flavonoids, most likely are metabolized. We really need to study the metabolites and see if they do have any in vivo activities to confirm this hypothesis, and it came back, this whole grant application came back with the comments, this is the stupidest thing we ever heard, this has no scientific merit. So this was 2008, meanwhile we have 2016, and meanwhile all the funding agencies are very interested in studies like this, because you have this discussion about the human microbiome and how it interacts with nutrients and metabolizes compounds, and that it is very important to study the activity of those metabolites. Well, at that time, what are you going to do? You need to do research, but you don't have money, so you do studies maybe on a broader perspective. You try to find new funding for new projects, and so we went beyond the usual proposed bioactivities of flavonoids, because we knew it is necessary to study the metabolites, and so we thought, okay, is there anything else what flavonoids maybe could do? And so we had this hypothesis, maybe they can be used as biogenic additives and maybe therefore improve the biopharmaceutical properties of poorly soluble drugs. Now, I'm going to explain this term biogenic additives. It comes from a different, comes from a broader perspective. Actually, usually when you work with a plant extract, you have active compounds that are responsible for the proposed activity, but on the other hand, you also have so-called co-effectors. And here you can differentiate between so-called adjuvant compounds, and here I would like to introduce the term biogenic additive, but on the other hand, you also have fibers. Now, what can they do, these biogenic additives? So they can improve the biopharmaceutical properties of compounds. They can either stabilize the compounds or maybe lead to a solid dispersion. So just to give you an example, for example, saponins, for example, they decrease the surface tension. That leads to a better solubilization of compounds, for example, or you have maybe biogenic additives that through complexation lead to a better solubility. So I'm not going over this entire slide, but I think you get the idea and the concept what flavonoids might do. Now, we took, to prove our hypothesis, or to evaluate it a little bit further, we used as a model compound hypericin from Hypericum perforatum, St. John's wort. And I think this was a bad choice that we did, because this is the most difficult compound to study at all, because if you look at the structure, this molecule is I mean, it's flat, it behaves like a rock when you put it into an aqueous solution, and it's not soluble at all. So the estimated water solubility is around 6 nanogram per liter. So it's a very complicated molecule. And also, if you try to determine the log P value, we use the computerized log P value, it's around 8. So compounds that have a good oral bioavailability should have a log P value around between 2 and 4. So 8 is way beyond. So it's the absorption. You can predict that it's very difficult that this compound ever gets absorbed. And if you look at the polar surface area, you see it's 156 angstrom. And, you know, hypericin is supposed to be one of the active compounds in St. John's wort responsible for the antidepressant activity. That means somehow this compound needs to get into the brain. The compounds that have a chance to enter the brain need to have a polar surface area below 90. So 156, very difficult probably. Now, these were the biopharmaceutical properties that we determined, or that were determined actually. You also can look these numbers up in the literature. Still, we decided to use this as a model compound. Now, this is older work that we did. It was part of my PhD that I did, so what we at that time were interested in, if hypericin, the water solubility of hypericin can be improved if you add other polyphenols, in this case epicatechin and also procyanidins, B2 and C1. What we could see is, especially procyanidin B2 could significantly improve the water solubility with increasing amounts. And if you add too much, then the solubility decreases again. And so at that time we were also interested, okay, increasing the water solubility is one point, but can you also improve the bioavailability? And actually, what we did is we simply gave hypericin alone and then in combination with procyanidin B2, and could see that if you give this combination, you detect more of the compound in the blood. This was also a rat study, actually. And we said, okay, fine. Are there other polyphenols which also improve the water solubility and then therefore the bioavailability, and what's the mechanism behind this? So Now, what we did is we checked, we used some model flavonoids, and here we used quercetin as an A-glycan, quercetin with a rhamnose here in position 3, and then rutin with a double sugar glucose rhamnose also here in position 3. What you can see is when you only take quercetin, the A-glycan, okay, the water solubility improves a little bit, but best effects you can see if you use quercetin, and this was also a little bit better than rutin here with the glucose rhamnose in this position. So we said, okay, quercetin, this is our model compound, and we did transport. We determined the transport characteristics of hypericin in CACO2 cells. What you can see here, so red is hypericin alone, blue is in combination with quercetin. And these are the transport studies in CACO2 cells. So you offer the compound apically, and then you detect you try to detect the amount on the basolateral side. And theoretically, how it works is it should disappear here on the apical side, but you would expect if it's transported relatively well, you would see an increase here on the basolateral side. But you see a decrease and not really an increase. A little bit for the combination, but this is really not enough. So the question is, where does this compound go? So first we thought, okay, maybe it's a stability issue. Maybe the compound is not stable in this transport medium. So we checked the stability, and yes, you see a little drop, but this drop does not explain actually this disappearance of hypericin on the apical side. And then we said, okay, let's do recovery experiments, and we checked the amount of hypericin on the apical side, basolateral side, you see a little bit when you give the combination. But interestingly, a large amount of hypericin sticks to the cells. And this is actually now really a surprise because it's a very lipophilic compound, actually. And a little bit also sticks to the plastic. And then we compared the amount of hypericin after cell extraction, hypericin alone, and then in combination with quercetin, and this amount was significantly increased. So obviously a lot sticks to the cell membrane or is transported into the cell. So we did a detailed mass balance study, and this actually confirmed this. So you have, when you give the combination of hypericin together with quercetin, you have a little bit is transported to the basolateral side. In comparison, when hypericin, if you give it alone, nothing is transported to the basolateral side, and larger amounts are transported into the cell or through the cell membrane, actually. Now, then we asked the question, okay, quercetin as a model compound, that's great, but are other compounds, do they have the same effect? So we tested isoquercetin, hyperoside, rutin, and quercetin, and we didn't really see a difference. So quercetin still was the best, actually, compared to all other flavonoids. And then we did double combinations, triple combinations, and even we gave all combinations together. And there was not really, we couldn't really say that the more flavonoids you combine and give it together with hypericin, the better are the transport characteristics. It actually remained unchanged, and we couldn't really observe a pattern. So then we said, okay, fine, flavonoids is one point, but what if we work with extract matrix? If you use an hypericum extract, just dilute it a little bit and only have the matrix, and then do the transport experiments. And then you could see this significantly improved. Again, it decreases on the apical side, but, and this is, maybe you cannot see it here, but this green point here is the appearance of hypericin on the basolateral side in combination with extract matrix. So the more you give, the better the transport characteristics actually get. And this is, again, the detailed mass balance study, where you could see that if you give it together with extract matrix, you really have an improved transport. And again, also here, this is the amount of hypericin detected after cell extraction. And you see dose-dependent effects when you give extract matrix, that you can see an increased amount of hypericin. So just to summarize this part, quercetin seems to be a very ideal flavonoid for the transport characteristics. We couldn't really see synergistic effects. And what is still unknown is if there are other compounds in the extract, maybe phenolic acids or so, which also increase the permeation characteristics. Now, what has this to do with our original hypothesis, flavonoids as biogenic additives? So we are not the only crazy people, which is good actually. So there were other groups having the same idea, and they didn't, maybe they used easier molecules actually. They used, in this case here, synthetic molecules. Here even they worked with different antibiotics, and they also came here to the hypothesis that if you use flavonoid cocktails, this would be a potential approach for multi-drug resistant reversal in, for example, cancer treatment. So I think this really is valid, and there's something that I think should be further employed. Do I still have time, or am I running out of time? 5 minutes. So I will be really quick. I won't get everything done in 5 minutes, but I try to speak even faster. So now, one of the challenges in flavonoid research is actually, and I mentioned this at the very beginning, is the large-scale production of flavonoids. And usually, again, if we remain to this hypothesis, flavonoids are the magic drugs, we would need a lot of them. But where do we get them? Today, mostly the production relies on isolation from plants. And if you do a chemical synthesis, you require extreme conditions and very toxic chemicals. So this is definitively not environmental friendly. You can use metabolic engineering and synthetic biology, but still you will not get the amounts that you would need. And I'm not speaking of milligram amounts, I'm speaking of tons, and that's really a large amount. Now, are there alternative sources? And in the last couple of years, I was working on a project involving Rosa damascena, the oil-bearing rose actually. And here you just very quickly how rose oil is produced, and then I'm showing you what the relevance for this is. So the flowers are harvested, and they are brought immediately to the distilleries, and usually I should mention that Bulgaria in Europe is the largest rose oil producer in the world, next to Turkey, and the flowers are collected, they are immediately brought to the distilleries, then they are here thrown into these tanks. You do the water steam distillation, and the final product is the rose oil, which actually goes in all perfumes, and like companies like Chanel, Dior, they all use the rose oil actually. And something that probably you might not be aware of is for one distillation cycle, to produce one kilogram of rose oil, you need approximately 500 to 1,000 kilogram of rose petals, of the flowers actually, and 2,000 to 4,000 liters of water. You have one distillation cycle, 100-degree hot water, and one distillation cycle takes 2 hours. And what happens afterwards? Okay, you get your nice oil, but you produce large amounts of waste. You also have the solid waste from the flowers, but you also have a large amount of liquid waste. Annually, that's approximately 30 million liters of wastewater that you produce. So this is the river, how it looks before the distillery, and this is the river, how it looks after the distillery. What you see here, this is not soil, these are the distilled rose flowers actually. So basically all the compounds are extracted here. And what you see here, this water is the wastewater from the rose oil production. And you see it's steaming hot waste. At least it doesn't smell, it smells nice, but it's still a really serious environmental problem. Our idea was, okay, can we recover the polyphenols? First of all, what's in this liquid waste? We focus on the liquid waste because from the amount, that's the largest amount actually. We tried to develop a method to recover the polyphenols. We used adsorption resins actually, and I can make this really quick. So we upscaled the process, and we detected that in these liquid waste actually are many flavonoids, especially quercetin, ellagic acid, and all those derivatives. And then we also wanted to know, okay, does this concentrated waste that we recovered, does it have any activities? And knowing that it contains flavonoids, we didn't go for any oral applications because we knew it's difficult to formulate and it's difficult to find an appropriate product because you have all these problems with metabolization and so on. So we went for topical applications. And we found that these polyphenol-enriched fractions has strong antiproliferative activities. It decreased the levels of vascular endothelial growth factors, so it decreased actually growth, it induced growth inhibition. and also inhibited cell migration, as you nicely can see it here. So this would be our positive controls. This is our control including FCS, which we used as positive control. These are our positive controls. And as you can see, quercetin here, but also ellagic acid, the extract and especially here one fraction had significantly inhibition on cell migration. So that we think, and this is actually almost my last slide, that we think such a polyphenol-enriched fraction had very potent inhibitory effects on cell proliferation and cell migration, and therefore might be interesting for further development for the treatment of hyperproliferative skin diseases, for example, psoriasis, for example. And now our approach actually using this agro-bio-waste could therefore maybe could close a gap in the raw material supply chain, because this byproduct might be really, it's easily accessible and could be further developed. And as a conclusion, I think I showed you two projects that, in my opinion, are very interesting. Flavonoids as biogenic additives, which I think can be further developed. And of course, I just would like to focus a little bit on that agro-bio-waste might be an important source for flavonoids, and this would enable us for further large-scale production. And now I'm coming to my last slide, and this is thank you to all people who were involved in these studies, and especially also the funding agencies. Thank you.